Hello, welcome to the MalariaGEN Podcast, using genomic surveillance to better understand how to control and eliminate malaria. MalariaGEN.net. Hello, today I will be speaking to Professor Olivo Miotto, Associate Professor at the Nuffield Department of Medicine, Oxford University. We'll be talking about the GenRe-Mekong project, the largest implementation of molecular malaria surveillance to date. So welcome, Olivo. Can you introduce yourself and tell us a little bit about your research, please?
Good morning, Heather. It's very nice to be here. My name is Olivo Miotto, and I am a researcher working for Oxford University, based in Bangkok at the Mahidol Oxford Research Unit. I specialize in the genetics of malaria, in particular, the genetics of the parasites that cause the disease. Malaria research is really divided between people who study malaria in humans, malaria in mosquitoes, which transmit the disease, whereas I, like my colleagues, actually study the parasite that causes the disease and lives in your red blood cells. And what I focus on is collecting blood samples from malaria patients so that we can read the genetic code of the parasites, read the DNA. And if we can do that on many people, on sort of large population samples, then we can compare the genetic codes of different parasites, and we can look at variations that may result in sort of changes of how the parasite behaves. So this is what my focus is. We call this genetic epidemiology because we are looking at how these changes are distributed over time and over space. What we do apply this on is what we call genetic surveillance. In other words, we are looking at patterns of changes in the genetic code of the parasites, and we are mapping them over the territory, and we are trying to predict what the characteristics of the parasites in different parts of different countries, what they're like, and for example, whether they are resistant to the drugs that are being used to treat the disease. And that's a very important problem in malaria. So this is what I do. I don't know if I made it sound easy or complicated, but it does require a lot of people to work with me and my group in many different countries.
So picking up on that, Olivo, can you tell us briefly about the group that you're working with, GenRe-Mekong, and the ambition of that project?
So yes, the GenRe-Mekong project was started about 5 years ago. It is funded by the Bill and Melinda Gates Foundation. What we are trying to achieve is to engage with the national malaria control programs of various countries in Southeast Asia, in what we call the Greater Mekong Subregion, which goes from Myanmar to Thailand, Laos, Cambodia, and Vietnam. We want to work with the control programs in these countries in order to sample blood from patients in the areas where malaria is endemic and inform the control programs about the genetics of the parasites in a way that allows them to respond in time and basically change their interventions if it's appropriate. For example, a very big problem in that region is the emergence of drug resistance. In other words, the parasites are developing mutations that make them less prone to be killed by the drug that's being used to treat the disease. And this has happened repeatedly in Southeast Asia. What happens is that these resistant mutations tend to spread. If they get to Africa, where most of the deaths from malaria happen, then this is a big disaster because basically the drugs become ineffective. So we are trying to map out where these resistant parasites are, and we're trying to contain them, and we're trying to eliminate them before they can spread out to where they're going to do maximum damage. We started off firstly by working with some of the big research projects in the region that were working at different sites in different countries. So we started mapping out the resistance to the main drugs that are being used in this region. And gradually, we also then engaged with the national malaria control programs. And right now, we're working with the governments of several countries in the region and putting them in the driving seat when choosing where to get the samples from and how to design the surveillance network. And we do all the technical work, the processing of the samples, and produce results that they can use in order to respond to the changes that we're reporting. The project over time has transitioned from one that involves mostly researchers to one that involves largely public health authorities. But a lot of the technical work does require the sort of research labs to be involved. And in particular, we have a very strong collaboration with the Wellcome Sanger Institute in the UK that does a lot, not only of the sample processing, but also of the computing aspects of the project. And the sort of very nice thing that's happening gradually is that we are transferring some of these analysis capabilities to local laboratories in the region so that the project will be able to be run more autonomously in these countries.
You talk about sampling over a whole region. What would have been the main challenges to sampling and genotyping these parasite genomes?
As you know, malaria is a disease that mostly affects rather poor regions. Just being in an area where resources are limited, it even means that it's difficult to transport the samples to and fro. It means that the people working in the clinics may be more of a challenge to reach to them, to work with them, to understand whether they have issues with the projects and so on. I mean, some of them don't even speak their own national language. They speak some local dialect. So it is hard to get to a large number of collaborators engaged and working to implement this in a homogeneous fashion. I'm happy to say that really we've had very good luck in that respect. It's probably not down to luck. It's probably down to the willingness of our collaborators to do a very good job. And so in spite of the sort of simple settings in which we operate, we do get very good performance from the project. To some extent, it was difficult to get started with the project because it's a very new technology. It's a very new capability that we're offering. And it was quite difficult for the public health authorities that we were, if you like, selling the project to. It was quite difficult for them to really visualize what value we were going to deliver to them. Therefore, it was very helpful to get started by collaborating with research projects that are a bit more familiar with the technologies, and then have some results to show to the control programs so that we could explain better what value that we're going to get out of the project. And this has worked very well. And now we have several of these control programs that are very engaged, and they participate actively in the discussions about the results and what they mean to them and how they should respond. Although, of course, the decision process is really up to them. What we are trying to do here is just provide them with the maximum information that will help them make that decision.
How have you simplified the process? I believe it's called Spot Malaria. to make it a more stable and sustainable way of collecting samples?
I think in many ways, our biggest struggle when we started off was to, as I often say, was to not be a pain to these people. In other words, how to implement a project where we profile genetically as many parasites as we can from a country without actually disrupting the clinical operation in areas where it's already quite difficult to conduct these operations. So what we did is we really made it as simple as possible. And right now, what one of these health workers at the clinics has to do in order to collect a sample is basically to get a drop of blood from a finger prick on the patient and put it on a piece of filter paper that we provide, dry the filter paper, put it in a little plastic bag with some silica gel and keep it there until somebody comes and collects it. And of course, they have to give the patient some information about the project and make sure that the patients are happy. But overall, we've made the process as simple and as easy to integrate in the normal routine of the clinicians as possible. So this has, I think, has been a very big element to our success, because it has enabled the control programs to really sort of spread out over the territory, even to the most remote places.
So you have samples coming in from a whole region. How is that analyzed to detect drug resistance?
Yes, there is a number of ways in which we identify new drug resistance. We could call them strains or variants, if you like. We become quite used to that word in the last couple of years. We are most interested in drug resistant strains. We don't necessarily do the detection of drug resistance ourselves. Sometimes this is done by clinical studies instead. There may be reports that drugs are failing or that they're taking longer to act than is usual. And the important thing for us is to identify what variants, let's call them that, are causing these changes in the phenotype of the parasites, and then we can track these because we can get their genetic signatures, very much similar to what people have been reading on the newspaper in the last couple of years about alpha and delta and gamma and omicron strains, that they're characterized by different mutations. That's how we know that you have a delta versus an alpha virus. And similarly here, we are able to look at different mutations and identify which parasites belong to which strain. And therefore, we are able to track the strains as they move across the territory. And again, so it's a fairly similar process to what epidemiologists have been doing with the COVID virus.
How is the data that's generated from that process used to monitor the spread of drug resistance across the region?
One of the challenges that we had was how to communicate these results back to the national malaria control programs and to the World Health Organization and other organizations that can benefit from these data. Usually, public health officials don't necessarily have a background in genetics. And even if they did, actually the sort of evidence for drug resistance markers and their significance is quite a sort of complex field that requires a lot of literature research and all this. So what we felt is that we had to very clear and understandable information to the control programs, and reporting that 20 parasites had the C580Y mutation in the calcitonin gene just wasn't going to do it. So what we do is we use the genetic data in order to make predictions. Instead of saying what I just said, we would say that, okay, in this particular district of this province, there is about 25% of parasites that we predict to be resistant to artemisinin, which is a drug that is used as a frontline. And actually, in order to get a visual of that, we produce maps, geographical maps, with traffic light colored blobs that show green good, red is bad. We show the prevalence of resistance to various drugs, not just artemisinin, across the territory where we are sampling. And it does appear that this is a very effective way of translating the results. And the control programs are using this. I mean, they feel this is an intuitive way of delivering the results. And they are using this together with other data in broader analysis, what interventions must be delivered across the territory.
How is the project working on a regional scale? How does that support a better policy at a region-wide level?
And what we have seen is that really national political boundaries really don't seem to matter very much to the spread of the disease. This was very much a case in point in the period between about 2016 and 2019, where there was this sort of very fast spread of one particular multi-drug resistant strain, which took everybody by surprise. And actually, the reason we know about this is because we've actually put together data from multiple countries. I believe now the control programs in the different countries do understand very well the value of putting surveillance data together from different areas and looking at the picture in a regional context. And the nice thing about the data that we are delivering is that they are delivered homogeneously in different countries. It's the same genetic report cards, as we call them, that are delivered in Laos, as they're delivered in Cambodia, in Thailand, in Vietnam. Therefore, putting it together in a regional map is really a very simple exercise. And it is an exercise of benefit to all the participant countries. So we are working now with not only with the malaria control programs, but also with the scientific experts that support them in the different countries to create a regional analysis group, so that we can all see these data, we can all understand what they mean, we can all follow the spread of these strains, and we can advise accordingly to the different governments. I have to say the attitude of the control programs is really great in this respect, and they've actually embraced data sharing in a very good way. I hope we can actually extend this in the future. It's really nice development to see.
So are there particular developments that you anticipate now in the coming year based on how the project is being implemented across a region?
Yes, we are currently in a very fast changing situation in Southeast Asia. In the last year or so, there has been quite a big drop in the number of cases. We don't know to what extent this might be tied in with the consequences of the COVID pandemic. Certainly, it is largely the result of very well implemented elimination strategies by the control programs. So it's probably a mixture of factors. So because things are changing, the epidemiology is changing as well. As the numbers reduce, paradoxically perhaps, the risk of new forms of resistance actually increase. And therefore, this is a time when we really must be very careful about surveillance. And what we are somewhat concerned about is that new strains and new forms may emerge, for some of which we may not even have markers available to monitor. So actually, one of the exciting things that we are involved in now is to, rather than look at markers of resistance, we look at either the whole genome or what we call genetic barcodes, which are sort of relatively simple, like summaries, if you like, of the genome that we obtain at the same time as we obtain the markers of resistance. And by comparing these, we can look at the similarities between parasites and their differences. And one of the things that we are doing now is to look at outbreaks and to see whether we think these outbreaks are just the result of normal transmission of malaria, just more mosquitoes, more rain, or whatever, so there are more cases, or whether they're driven by some specific strain of malaria that has some new capabilities. And we are developing some methods for doing this, and these outbreaks do tend to occur as the number of cases drops down. So this is a very critical time, what we call the pre-elimination stage, where new types of surveillance are needed, and new processes that may lead us to monitoring these strains, even as they emerge with new markers that we might not have validated yet.
What would you say have actually been the highlights of the project? They may be too many to name, perhaps.
There have been really many highlights. I would say the most thrilling things that happened was to actually know that we had influence policy. You know, when we started this off, it was really a pioneering effort. Nobody had done this in malaria before. It was a big technical effort and a very big logistic effort. So to actually come to a point where, you know, we got a message from the control program saying, yes, thank you very much, that was very valuable to us, and this is what happened, was a validation of all that we had hoped for. And this has now happened more than once. And so we think we are really on a good path here. And we will strive to make this more and more useful and have more and more moments like that.
And I think you started when we began this conversation talking or hinting at knowledge and skills transfer. How do you see that developing over the coming years?
This is very much a question that's at the forefront of our thinking. How do you sort of make sure that participation in endemic countries isn't limited to just collecting samples and giving pills? And this is very much an issue with the sort of technologies that we're using, because they're often fairly expensive. They often require a lot of know-how, and typically know-how that is not easily acquired in endemic countries. It's more the domain of sort of Western universities and companies. We have been working on how to package these technologies so that we can start implementing them in local laboratories in the countries. So that's one aspect, you know, like when, you know, taking the blood spot, and instead of sending it to Europe to be analyzed, you send it to a local lab, and we generate data. But often the stuff that's thin on the ground in these regions is actually the informatics capabilities, the computing power. So we are actually providing cloud services to the countries that they can use in order to process the data. And again, it helps a lot to have a very standardized method, because then it means that we can all use the same tools, whether the sample has been processed at the Sanger Institute or it's been processed in Vietnam or in Thailand or in Africa, of course. I mean, we are actually working with African groups to do the same things in that continent too. We are transferring technology and we are supporting with shared tools. And whilst we realize that it will be hard to have the same level of technology available in all countries, I think it is important that we develop the skills of the next generation of scientists in the endemic countries, so that the countries can be more self-sufficient in running their own programs. But also, it is just generally important for science in these countries to have the right skills available.
Thank you, Olivo, for taking the time to be here and explain a little about the successes of the GenRe-Mekong project. I look forward to hearing more about the next phase soon.
Thank you, Heather. It's very nice to be here. Thank you.
Bye for now. Brought to you by MalariaGEN.net.